Hi everyone, welcome to Molecular Biology and Biotechnology with Lucy, the educational platform for all your basic molecular biology and biotechnology. Thank you for being with me all along. I really appreciate your support. And if you're new here, welcome. And remember to hit the subscribe button so that you're also notified whenever we post a video. So today we'll be looking at vectors, an introduction into vectors. We shall do it in a series. So today is just the introduction, and then we'll go on into the different types of vectors. So we mentioned vectors in a previous video, which I'm going to link up here on the recombinant DNA technology steps. And the very first step is where you obtain your target DNA and your vector that is going to help you carry your gene of interest or your target DNA. So usually there are many types of vectors, but this case we are talking about those that are in molecular cloning, because we have other vectors such as, organisms that transfer or transmit infections such as mosquitoes. So in this case, we are talking about vectors in the context of molecular cloning or molecular amplification. So a vector is a DNA molecule that is used as a vehicle. So we use it as a vehicle to carry artificially foreign genetic material into another cell. So you have now your target DNA fragment or gene. For example, if you can use the situation of COVID-19, we have the gene that codes for spike proteins. So that DNA fragment will be inserted into a carrier, which is our vector. And this usually results into what we call recombinant DNA technology. And therefore, this vector usually will help us to carry it into another cell. And in this case, the other cell is the human host. And here, once it reaches the host, it can either be replicated or made into many copies of, or it can be expressed into RNA or into proteins. Now, a vector, of course, must possess certain minimum qualifications to be an agent of transfer, of maintenance and amplification of that target DNA. And we shall be looking at some of these qualifications in another series. So let us reflect on the four main functions of a vector. So one of them, which is the most basic, is cloning. And cloning comes from the word clone, and clone means making unlimited copies of something. So in this case, we have inserted our DNA, we have stored, and it can be manipulated in the vector. So the vector is going to help us produce unlimited amounts of that particular fragment, as many copies as possible of that particular fragment of interest. That's a cloning vector. Then we have what we call an RNA transcription vector. And from the word, it also means we are making RNA through the process of transcription, because we have cloned a DNA insert, made many copies of a DNA insert, and now we want it to be synthesized into high yield, good quality, full length RNA. And that is usually the process of transcription. Okay. Then we have another vector that we call protein expression vectors. And in this one, even from the word, we are simply saying we are able to make proteins from our vector. So we usually make sure that the vector is constructed in the way that whatever DNA molecule we insert, we put it between a promoter and a terminator sequence. That is the beginning of transcription and the end. These ones signal the beginning and the end of transcription, so that we are able to have the transcription process into an mRNA and then finally translated into a protein. Usually the process is DNA. We replicate the process that happens in living organisms, and then it goes to RNA, and then we express proteins. So this is now what we call transcription. this process of transcription, and this is what we call translation. So for us to have proteins, we have to have first transcription into RNA, then translation into protein. So we have vectors specifically used for this purpose, and that means they must have a promoter sequence, they must have terminator sequences and other characteristics, which we will be looking at. Then we have another vector called a shuttling of DNA vector, and to shuttle means to transfer from one organism into another. So usually these are vectors or plasmids that are capable of propagating and transferring genes of interest between two organisms. It can be between a prokaryote and a eukaryote. So some of these plasmids or vectors usually will have those from E. coli and Agrobacterium to Mephistians. So for example, for this Agrobacterium, usually it's a soil bacterium, and it will infect our plants where we have woods and cause tumors. So we usually are able to use this one to transfer small segments of DNA into a plant genome. So if you want to transfer a certain DNA target or fragment into the genome of a plant, then it is possible to use this Agrobacterium to Mephistians in a process called transformation to be able to transfer your gene of interest. So that is what we call shuttling. So it's able to transfer. You need this DNA inside another organism, then you use a vector or a medium or a means of transport or transfer or vehicle that can be able to get your DNA from that one organism into another organism. So those are the four major functions of vector: cloning, RNA transcription, protein expression, and shuttling of DNA. Now, based on the nature and where they come from, that is their source, vectors can be grouped into bacterial plasmid vectors. We have bacteriophage vectors. We have what we call phagemids, cosmids, viral vectors, which can be animal viral vectors or plant viral vectors, shuttle vectors, yeast, artificial chromosomes, and transposons. So in our next series, we will be looking at each of them in details. So coming up, we'll look at the cloning vectors, each of them, what are their characteristics, as well as expression vectors, each of them, and what are their characteristics. Yes, that is it for today. Thank you for watching. Remember to like, to comment, and to share, and to subscribe if you have not subscribed.